It's a pleasure to.
Thank you. So I'm going to start now. I'm sorry. So I will present you today how to use isotopes to trace trophic interactions in symbiotic organisms. First of all, I wanted to make a small presentation. So I'm coming from Monaco, from the Principate of Monaco. Previously we were in the Oceanographic Museum, and now we moved to new facilities which are here next to the harbor, and we are just on the top of the building. So we have also water coming to the top of the building. So the water is pumped in the sea, is brought up to the roof and goes down to our aquaria. And so we work on corals, so we have a big setup, big tanks with aquaria inside. And so the water is coming directly, it's an open flow system, and we have liters of water coming every minute to the aquaria. These are the main aquaria. Then we have also small rooms with small aquaria where we can change the light, the chemistry of the water. We can change the temperature and everything. And so we can do ecophysiological or ecological experiments. So my team is made of 3 researchers, 2 postdocs that are with me for 3 years now, 1 PhD student, and I have really 2 technicians. These 2 are more for the whole institute, doing things for everyone. And as Ulysse has told you, we work on 3 different types of corals. We work on the tropical corals, which are mostly in the tropics and subtropical areas. We work on the Mediterranean or temperate corals. So, no, we work, on the cold water corals coming from 200-300 meters depth or 500 meters depth, which are mostly located in the cold sea, in the cold seas here, in the North Sea and the Atlantic Sea. And we work on the Mediterranean anthozoans, I would say, corals or gorgonians, and it's the yellow part here. And why we have these 3 different corals, it's because the tropical corals, they form reefs, and they live under stable temperature and light, but usually it's high temperature and high light. And they are almost symbiotic with algae. They cannot, most of them, those who are building the reefs, they live in symbiosis with algae. Then we have the temperate anthozoans. They mostly, well, they don't form reefs, and they live under variable temperature and light conditions. So we have different, in terms of ecophysiology, we have different conditions compared between these 2 corals, and they can be symbiotic or non-symbiotic with algae. And finally, the cold water corals, they form also big reefs in the North Sea, as you can see here. Here is the diver, and this is the reef. But they live under dark conditions and cold temperatures, and they are non-symbiotic. So we have all, working on these 3 different corals, we have all conditions of symbiosis and all conditions, environmental conditions of temperature and light. And so the goals of the team, we have several goals. As we talked, we want to assess the coral response to global change or pollution or eutrophication, everything, plastics also, contaminations and things like this. We would like to identify early warnings of ecological disturbances. We also work on the nutrition, the nutritional interactions among the different partners of the corals, and we do some paleoclimatology studies, looking at isotopes and trace elements in the skeletons to reconstruct past climate. And today I will just talk about the nutritional interactions, just one subject, otherwise it would be too long. So why we should study coral nutrition? Because this is more for the students, but nutrients are the building blocks of the proteins, of the amino acids, of everything. And thus the acquisition of nutrients is important because for coral growth, which will determine the reef expansion, for the coral reproduction, which will determine how their ability to colonize new environments, and also for the coral health. And this will be with the resistance to stress, because a coral that is well fed, it's like us, will resist better to environmental stress than non-fed or corals in poor conditions. And so what is a coral? Because no one is mostly working on corals here. So corals are an association of a lot of tiny organisms like this, which are called polyps. And the polyps is like jellyfish, so like the many of you who are working on jellyfish. So they have tentacles like this, just a stomach, and they can secrete a skeleton. And all the tiny polyps all together living forms a colony. And within the tissue of the corals, at least for the tropical ones and some Mediterranean ones, we have algae colonizing the tissue of the polyps, and we can have 9 different clades. They haven't been really determined until yet. Well, they start to be determined, but they have quite a lot of different algae inside the polyps. And they also have microbes, such as viruses and bacteria, which can be in the tissue, in the skeleton, in the mucus layer of the corals. And so we have these three partners, and since we have these 3 partners, it's called an holobiont, because it's several organisms making one organism together. So we have the animal host, the algae, zoops or dinoflagellates if you want, and the microbes. And in terms of nutrition, all these different organisms will feed independently, I would say. So the animal host, it's like a jellyfish, so it will prey on plankton, organic nutrients, and get the C and N directly from the prey. Then the algae, they will, as all algae, use the inorganic nutrients such as ammonium, nitrate, or inorganic carbon, phosphate, and through the photosynthesis will transform these inorganic nutrients into organic nutrients. And for example, we have the microbes. Here I just summarized with the isotopes. So because it's mostly the only microbes for which we know their function, I would say. They have plenty of the microbes, but we don't know at all the functions of these microbes. For the isotopes, they can fix the D-nitrogen, N2, and transform this D-nitrogen into ammonium. And since we have an holobiont, all the nutrients are continuously exchanged between the different partners of the symbiotic association. And of course, every flux will change with the environmental conditions. So, I mean, if there is no, if the conditions are not favorable for N2 fixations, therefore this flux will be shut down compared to the other flux. What we know in corals is that the algae are, through the photosynthesis, are bringing 90% of the energetic needs of the symbiotic association. So in normal conditions, most of the researchers think that the algae are supplying most of the nutrients to the corals, 90%. Except that now, due to thermal stress, I don't know if you have heard about coral bleaching. coral bleaching is just because the brown color of the corals is due to the presence of algae inside the tissue. So due to thermal stress, the corals expose the algae and they become white because the coral tissue is white. And then they have to rely on other nutrition modes because this one is shut down. And bleaching is quite important. Here you can have an entire reef completely bleached after warming. Here also, except these guys here. And usually if the bleaching is too intense or it's too severe or lasts for too long, then you can have all portions of reef completely dead because they lose their main source of feeding. And here, just for 2019, this year, all the red dots here are locations where temperature has been above the natural temperature and which have induced bleaching, coral bleaching. So, to study the nutritional interactions is very difficult because we have several partners that exchange continuously nutrients from one to the other one. So it's difficult to know who is benefiting what. It's also difficult to know who is getting what. And most of the nutrients are also lost for the respiration, the mucus release. So it's a bit a nightmare to look at the energetic budget of a coral species. And it's why in the lab we use isotopic labeling of the food to try to trace this labeling inside the coral tissue and inside each partner. And I will give you today 2 or 3 examples, depending if you are hungry or not. So the first example is with the heterotrophic nutrition of the host. So here, so how much of the host is getting food from the plankton, from outside. If you're lost, you tell me, you just stop me and I can get questions because it's not easy. And one of the main questions concerning this heterotrophy is to know if the heterotrophic acquisition of nutrients depends on the autotrophic acquisition of nutrients. Mean how the two are related each other. And usually, the general belief is that when the coral cannot anymore feed on autotrophy, for example during coral bleaching, for example, then it shifts on heterotrophy. And there is a balance like this between autotrophy and heterotrophy. So this is the general belief, and this is the hypothesis we wanted to test. If indeed this flow is increased when this flow is decreased in corals. Okay? So we made an experiment. This was my PhD student Leila, who published this year. So we tried to modify the fluxes of autotrophy, the fluxes that goes through the algae. So we made a condition where there was no autotrophy, and this condition was made just by removing nutrients from the water, removing the nitrate, the ammonium, the phosphate, just cultivating corals under nothing, I would say, in the water, no nutrients, no inorganic nutrients in the water. So the corals were quite white.